a short presentation in Italian. Quindi benvenuti a tutti e questo è il primo appuntamento del nuovo ciclo di eventi online di SoIPa che abbiamo, diciamo, delineato insieme sia a tutto il consiglio direttivo ma anche ai soci. Da gennaio, quindi questo è il primo degli eventi di SoIPa Webinar. Da gennaio avremo tutti i mesi almeno due eventi e abbiamo inviato il calendario a tutti ed è presente anche sul sito. Gli appuntamenti sono diversi, sono suddivisi in gruppi, nel senso che avremo il ciclo di seminari di parassitologia molecolare organizzati principalmente da Sapienza, che ormai ha una lunga tradizione ed è patrocinato SoIPa. Abbiamo poi il ciclo di Spring Parasitology, quindi quello dedicato ai giovani, non soltanto dottorandi, ma anche giovani ricercatori. Abbiamo giornate dedicate invece alle giornate mondiali, soprattutto per alcune parassitosi chiave, quali le Neglected Tropical Diseases, quella del 30 gennaio in presenza presso il Negrar di Verona e il 3 febbraio invece ci sarà un webinar dedicato da parte appunto del giovane gruppo SoIPA per le Neglected Tropical Diseases. Poi avremo le giornate dedicate a Chagas, le giornate dedicate a malaria. E poi abbiamo un altro pilastro che è rappresentato appunto da SoIPa Webinars, ovvero i webinar che sono suggeriti dai nostri soci oppure che sono stati diciamo ideati e pensati dal consiglio direttivo. E quindi il nostro attivo socio Marco Lalle qualche tempo fa, qualche mese fa, ci ha proposto appunto questo seminario e pensiamo che sia molto importante anche perché le cost action, questo sarà un primo appuntamento, ma ho intenzione di farne altri sulle diverse cost action che interessano i soci di parassitologia perché sono delle ottime opportunità di networking, soprattutto per i giovani ricercatori. E quindi abbiamo il calendario, questo lo dico ai soci, da gennaio fino a luglio, però qualsiasi altra iniziativa che possa essere proposta dai nostri soci, ben venga e siamo ben contenti di promuoverla. E quindi I switch in English. Tassos, thank you very much for your presentation that will be in few minutes on blastocystis and in particular on the cost action you have on blastocystis under One Health. And for a brief introduction, I give the word to Marco Lalle, who is going to present our guest and also the topic of this webinar. Grazie mille.
Thank you, Laura. Thank you to our SoIPa president, and it's my pleasure to introduce you Dr. Anastasios Tsaousis, Tassos for friends. He's a senior lecturer in molecular and in evolutionary parasitology at University of Kent. He graduates in biology at University of Crete. He did his PhD at Newcastle University, focusing on mitochondria of microbial eukaryotes. Moved to Nova Scotia in Canada for a postdoc at Dalhousie University in Halifax, working still on mitochondrial pathways in anaerobic protist. In 2013, he joined the School of Bioscience at University of Kent, and now Tassos is the principal investigator of the Laboratory of Molecular and Evolutionary Parasitology. His current research focus on investigation of the adaptation of microbial eukaryotic organisms, particularly Cryptosporidium, Blastocystis, Naegleria, Gregarines, and Ciliate, and their course in parasitic evolution and diversity. Tassos is currently the mind behind the cost action CA 21105, Blastocystis under One Health, started in May 2022. The main aim and objective of this action is to shape and direct a state-of-the-art and dynamic blastocystis network linking academia, health sector, and industry in order to enhance, spread, and apply knowledge and expertise on this gut microbe and its role in health and disease. So please, Tassos, the floor is yours, and thank you for sharing this important cost action news on blastocystis.
Brilliant. Thank you very much for the invitation, and thank you, Marco, for the introduction as well. I will share my screen first. Um, are we okay with that? Can you see my screen?
Okay, yes.
Yes. Brilliant. Okay. Hello, everyone. Today I'm going to talk about blastocystis, and I'm going to share basically some basic information of what we are doing on blastocystis, and we'll present it of what my lab has been doing over the last few years. And this will pass the ball to the course action that Marco mentioned as well. And just to understand what we basically don't know about blastocystis and what we would like to do. And basically, if I'm a good seller here, try to get you to be participating or be involved in our action as well. So I choose this title, Putting Blastocystis on the Spot, because I'm going to show some of the research we've been doing about blastocystis and what we know about blastocystis. But so how I structure my presentation, first of all, I'm going to introduce my lab. I think Marco mentioned already some of our research interests. I will give some background information on what is known about blastocystis. And this is the organism that you can see here on the screen. Then I will present some One Health case studies that we've done in Thailand, and this actually provoked us to basically build this cost action. We present some case studies in the UK because this is where I'm based and what we are doing. And then I will present the big questions about blastocystis and how the EU cost action is involved in that. So I will go very briefly on that because Marco already mentioned what my lab is doing. We are focusing on the adaptation of microbial eukaryotic parasites and their course in parasitic evolution and diversity. Sorry, to accomplish this, we are generating both genomics and transcriptomics data. We work on the microbiome as well. This could be either gut or soil microbiome, for example. We do metabolomics. We basically try to see what is being generated out of these microbes. But we also interested in the cell biology and the biochemistry of these organs as well. And basically, this is to see how they are adapting these microbes in living diverse and extreme environments. And I know it's this is a parasitology talk as well, and I talk about microbial eukaryotes of parasites because we still don't know whether some of these organs could be actually parasites or not. So Mark already mentioned that we are working with Cryptosporidium, Naegleria. This is my main pets, and of course, Blastocystis, which is the reason I'm here today. So, a brief introduction about Blastocystis. This is an organism that's been discovered more than 100 years ago. It was considered to be initially a yeast, and then later on it was recognized as Blastocystis. It went enterocola because it was something that was found in the gut. But later on, it was labeled as Blastocystis, got its own name, and it was almost now it's getting 40 years ago. Sorry, lost count. 30 years ago that Jeff Silberman actually put a home to this parasite and it belongs to Stramenopiles. And I will come back to that later because this is a weird position for blastocystis. So blastocystis has been found in the gut of individuals with and without gastrointestinal symptoms. So we don't know whether it's causing the disease or not. The big question is whether blastocystis is pathogenic or it's just an opportunist, and whether, if this is the case, are there specific species or strains or subtypes that could be correlate blastocystis with a disease. So, and I'm putting some papers out here just to give you an idea of what has been published over the years, they found blastocystis with people with gastrointestinal diseases, with diarrhea, with IPS or IPT, and they call it an infection as well. And I'm mentioning this because just because you find something in a patient with a disease doesn't mean it's always causing the disease. And these are cases that they haven't looked at anything else. They just said for blastocystis, and they immediately put a correlation, but we don't know whether this is true. And in response to that, this is a paper from Chris, from Ron Stensvold, that came out a few years back, and it's actually estimate that probably 1 billion people actually have blastocystis in their gut. So, and I think the number is getting even higher now with the advanced techniques of identification of blastocystis. So actually, 2 billion people having something that causes a disease might not be probable. And this is something we need to investigate even further. So if you go and do some microscopy on blastocystis, this is what you are going to see. This is a vacuolar form of blastocystis. It's a big rounded shape with a big vacuole in the middle, and you have the cytoplasms here with the nuclei and um, and this is the most transmissible form. So this was published. This is kind of the life cycle of blastocystis. It was claimed that there are a few forms of blastocystis. Some of them we haven't visualized in the culture at all, but we have the vacuolar, the amoebae, the granular, and our microscope is the form you will typically see.
Sorry, Sir Tsaousis, there are some problems of connection.
Sorry.
There are some problems of
Sorry.
connection. Yeah.
Should I close? Okay.
Okay.
The
Again.
can you hear me now?
Now, yes.
How about now? The videos.
Yes, the video, yes, but we don't listen you.
Sorry. Can you hear me?
Yes.
Okay, I will try to be a little bit okay. So I hope you and something I wanted to mention as blastocystis is actually
No, Tassos, we still have
and this is one of the reasons that research on blastocystis.
No. Tassos? No, I think you have some problem of internet connection.
Sorry, apologies for that. I don't know what's going.
I don't think it's
One
just a
second.
matter of Zoom. I think it's a matter of internet connection.
Check the microphone connection, please.
Yeah.
I'm going to try.
In progress.
Can you hear me now?
Yes.
Can you hear me now?
Yes.
Yes.
Okay. Okay, so I will try to use a different. I'm using. Sorry. I will use my iPad to talk. Can you see? And you can see my screen, right? Yeah.
Yes,
Okay, I
Tassos,
hope
but
this
there
is
is
working.
echo. There is echo.
Sorry. How about now?
Fine, fine.
Yeah. Okay. Brilliant. Okay. Sorry for that. I don't know what was wrong. Okay. So let's go one step back. Okay. So we have 4 different forms of blastocystis, which we can see under the microscope. We have the vacuolar form, the granular, the cyst form, which is the one found in the environment, and rarely we can see the amoebiform. But what is interesting about blastocystis is that it's an organism that is a strict anaerobe, so anything we can do in the lab is very, very difficult, and people working with blastocystis, they have a love-hate relationship because of that. And just to give you an example, if you actually leave it under 10-15 minutes on the slide, you will start getting a shriveling form of blastocystis, and at some point you will get the guts coming out, or whatever is in the cytoplazm coming out, and it's been bursting. So it's an organism that is very, very difficult to keep in culture. There are only 3 subtypes, 3 strains that are xenic. Most of them you can culture them with bacteria and they can survive a little bit, but this is one of the main difficulties of working with blastocystis. Anyway, I mentioned about blastocystis belonging to stramenopiles. Just to give you an idea, this is stramenopiles are here. They are closely related to alveolates and apicomplexans. Some of you are familiar with this group. A stramenopile's characteristic is basically, as its name says, is a flagella with hair. Okay, and this is a typical, what you see here is a typical stramenople, and you can see the little hairs under the EM microscopy. And obviously, blastocystis doesn't have neither a flagella, neither hair. And just to give you also an idea of what stramenopiles are, or heterocones, it's a very diverse group that includes the flagellates, the typical stramenopiles, oomyces, the kind of fake fungi, diatoms, and the giant kelp. So very diverse groups, and we have something that is completely reduced and not look like a stramenopiles to giant kelps that we can have cells that goes for a few meters. So a crazy group, one of the most diverse groups of eukaryotes that exist. Going back to blastocystis, it's been, nomenclature has changed, and we don't have blastocystis hominis, the one infecting humans. We actually have the blastocystis species because we found that most blastocystis can be found in all hosts. And currently there are 44 different subtypes of blastocystis, which infects basically birds and mammals. And this is based on the ribosomal RNA sequence. And 12 of the subtypes are found in humans. 4 of them, ST1 to ST4, are more frequently found, are more common. And the subtypes in different cohorts vary in prevalence. Distinct organisms like reptiles, amphibians, and insects have actually different other blastocystis other than the 44. And one of the questions it needs to be addressed is how variable the gene content of the genomes of the subtypes is. And I will come back to that in a couple of minutes. One of the things is being studied, and we continue to study within the cost action, is how frequently you can find each subtype in different hosts. For example, I mentioned that subtype 1, 2, 3, and 4 are most commonly in humans, with subtype 3 to be the most frequently found. The bigger the number is the most frequently found subtype in the host identified. In ruminants, for example, subtype 1 is the most frequent. In chickens is subtype 7, and in primates is subtype 1 and 2 and 3 coming later. The other interesting thing about blastocystis is actually geographic distribution. So as you can see here, in green, we have subtype 3, which is the most common subtype in Europe and in the rest of the world. But second, in actually Europe, is subtype 4, which we call it the European subtype, where in the rest of the world is actually subtype 1. And in Asia, it's subtype 7. So depending on where you are geographically, you might actually find a certain subtype more frequently found. If you look at the genome content of blastocystis between the different subtypes, ST1, ST4, and ST7 are the 3 subtypes that have been identified. So we don't have any bacteria in the culture, so we were able to do sequence their genomes, and you can actually see there are significant difference between the genome size, the GC contents of each of them, the nuclear coded genes, and so on. So there are huge difference between each of them. And just to give you an idea, this is comparison between the different subtypes. You see 1 to 4, 4 to 7, or 7 to 1. And you can see that between the genes, between the two, there are 10 to 20% difference between these two subtypes, or 14 to 20% difference here. And just to give you a perspective, the difference between Leishmania major with Leishmania infantile is only 1%, which are different species, or between C. hominis and Cryptosporidium parvum, Cryptosporidium hominis, is 2 to 1%. So we are actually having a higher difference between subtypes than there are between species in other parasites. So this is another weird thing about blastocystis. And the function blastocystis, you can see that you have a lot of genes encoding for cellular process signaling, metabolites, but we have more than 10% of its genome also is poorly characterized. And within that, we have some proteases, so it could provide clues that maybe there are virulence factors associated with blastocystis. Sorry. So just to summarize on the genome, there are considerable differences between the 3 sequence subtypes for a number of genomic features like DNA-based composition, size of genome, number of genes, and number of introns. There are organelles that we kind of be able to characterize them. For example, the mitochondria-related organelles, which have anaerobic metabolism. There is no peroxisome. There is some glucan metabolism, and there are some good repertoire of membrane trafficking machinery proteins. We have subtype-specific gene family expansion, for example, for peptidases that may relate to virulence. And blastocystis appear to have the most of the genes necessary for sexual reproduction. So actually, blastocystis can do sex. Another interesting fact about blastocystis is what I call it a lateral gene transfer magnet. Actually, more than we have identified already strictly 167 genes that have been acquired by lateral gene transfer, either from other eukaryotes, bacteria, or even archaea. And this is correlated to basically different functions and adaptations of blastocystis within the gut environment. And you might not think that the 167 genes are not a lot. It's actually 2.5% of its genome. So, and this is very, very strict observations. I think the number is even higher. So actually, blastocystis is able to acquire genes from the environment, from other microbes from the environment, to enable him to adapt in this extreme environment. And I can talk for an hour on the function of the genes. This is a representation that we have here, but I wanted to actually give an example of one of them, which this is alternative oxidase, or AOX, which is found in the mitochondrial lead organelles of blastocystis, and which we show that it's functional, it's been localized in the mitochondria, and basically we were able to see that actually blastocystis can deal with some accumulation of oxygen. It's not what's been considered to be a strict anaerobe. It might not. It may be able to actually deal with oxygen stress. And with that, we hypothesized that this might be one of the reasons that we actually find blastocystis in the gut with IBS and IBD patients. It might be actually be able to be there to help the microbiome, and because of this fluctuation, it's able to survive in the gut as well. And we'll come back to that later. So this was a brief introduction of what's going on blastocystis, but what we were missing, we have gaps on knowledge, on the biology, on the adaptations, even in the ecology of blastocystis in the gut. On prevalence, we don't know much of developed versus developing countries, rural versus urban localities, age groups, genetic background, longevity. All this information about blastocystis is missing. Transmission of blastocystis is being considered to be zoonotic transmission. Now the new figure from CDC is actually this one. It talks about fecal oral transmission, but could be the environment a reason of acquiring blastocystis? To answer this, we decided to actually investigate different settings, and it was easier for us to collaborate with my colleagues in Thailand, and we actually established the first One Health investigation in a rural area. We've done a tribe in school children, and we look at the water as well. And I will show you how all these are connected. So the aim of the One Health study was to actually investigate how blastocystis is transmitted in a rural area, in a village in Thailand. So we got in a small village near the borders in Chiang Rai. We collected samples from 45 humans, 44 animals, and from different water settings. And I'm not going to go through all this investigation, but we tried to do culture and microscopy, but we also extracted DNA to do both PCR and qPCR to see what is the most efficient method. And this is Vasana Jinatham, which was a PhD student working at that point on this project. you can see how many cultures she was been working on for 2 years to answer this question. So first of all, we've done a comparison between the different methods for blastocystis, and we've done different methods of blastocystis, and we were able to show that actually light microscopy was not that efficient for looking blastocystis. And when we moved to a more advanced molecular methods like qPCR, we can actually identify high prevalence of blastocystis in these samples. Among those, we actually found in all the samples, we found blastocystis in all the groups of samples, from humans to animal to water and in the soil. And we identified some novel subtypes for humans. For example, ST13 S23 was not identified before that. And we identified actually that some of these subtypes were also found in different environments as well. We've done some phylogenies to group them together. And what we actually show is that some of the humans or animals were grouping with environmental samples. So we decided to actually narrow this down further to look at how these are going on per household. So we explore blastocystis presence per household and to see whether they have animals as well. And what we actually realized is that there was not much correlation between positivity of certain subtypes with the animals present. So for example, just to give you an example, blastocystis, we found ST3 in humans, but ST7 in animals. Here, ST23 in chicken, ST7 in animals. So there was no connection there was no much of zoonotic transmission per household, which was a surprise, and it was actually questioning everything we had. But the answer was actually in the environment. So when we actually look at what's going on in the environment, we identified that some of the subtypes were actually circulating either in the soil and in the water. So this is the first time we actually had a circular conclusion on the transmission dynamics of blastocystis through a community and through a household to look at actually, for example, here given subtype 7 be circulating across all samples. And this was published back in 2021, and this was actually provoked me to put the course action, which we're going to talk later on. We did the same investigation in tribe school children in Thailand, and this was mainly not to look at the different age of the host, but also to see how the children are getting blastocystis, and with the animals surrounding them as well. And what we saw is that despite the fact that there were animals within the school area, there was, first of all, there was no correlation between blastocystis present or subtype present with ethnicity or weight or any metadata that we had. But what we actually seen is that blastocystis was, some blastocystis were stained. This is the first time we actually see longitudinal studies of blastocystis being stable colonized in a host, or it's been acquired. In the case it's been acquired is from subtypes that were not circulating actually in the animals, but actually in most cases it's been circulating within the water. So again, we've seen blastocystis presence coming up through another source than the animals. And when we saw that, actually, Vasana, the PhD student, decided to actually, sorry, I will come back to that later. So basically to summarize what we found so far, so these studies have revealed no clear patterns of direct transmission between human to human or animal to human in these communities. Instead, we have multiple independent routes of transmission. We've seen that comparison of sequence found in different hosts and environments indicated that highly similar strains of example SD1 and SD3 are circulating in the communities. This suggests that shared blastocyst transmission cycle amongst humans, animals, and the environment for these subtypes. And just to summarize, that we need an investigation and future investigation of blastocystis to be under an shared umbrella to look at everything, not only two different hosts, for example, to explore how this parasite or this microbe is transmitted. So, as I mentioned, the pattern we've seen, for example, this is from the latest investigation is we have water being one of the major sources of transmission. And Vasana, the PhD student in Thailand, she decided, without even telling us, to go and explore the water on different areas of Chiang Rai. This is, we are moving from a rural to an urban area. So she actually went in different places, collected water, and we investigated whether blastocystis was there or not. And what we identified is that we actually found SD3 be quite consistently present. And what was surprisingly is that in some of the cases that blastocystis was present, it was on sources where actually the water was chlorinated. And this provoked us to actually investigate the resistance of blastocystis under chlorine. And basically, what we've done, we explore blastocystis not only resistance to chlorine, but also whether if we expose it and reintroduce blastocystis, what will happen. And what we've seen is that typically concentration between 0.2 to 1 ppm of chlorine are effective to eradicate more pathogen. In the case of blastocystis, we actually saw 300 more time concentrations needed to actually eradicate blastocystis. So blastocystis is quite resistant to chlorine, and we want to actually investigate this even further to look at the mechanism of resistance. Excuse me. And this is just to give you an idea of the availability assays we've done on blastocystis that was resistant in chlorine. Moving on from all the kind of epidemiology and transmission dynamics of blastocystis, I wanted to introduce to the microbiome now, just to look at the more the ecological side of blastocystis. So there have been a few studies over the years looking at blastocystis in relation to microbiome, and some of them show association with richness of Prevotella or Minococcus or Bacteroides, lower Bacteroides, high bacterial richness, and so on. And in the most, I think, solid case, it was by Nieves Ramirez in 2018, where he actually showed that carriers of blastocystis have absence of gastrointestinal disease or inflammation and higher bacterial diversity. But all these cases were with only a few samples, a few dozens of samples. The biggest study on this was actually done by Italians. That's where I bring the connection. It was Nicola Seccata's group from University of Trento, where he actually showed that microbiome Blastocystis present is obviously associated with Prevotella as well, but it's actually show a more diverse microbiome, a much healthier microbiome. A continuation of this study came out this summer from the same group, Nicolas Secada's group in the University of Trento, where they've done 57,000 samples, and they show that actually Blastocystis is linked to healthier diets and more favorable cardiometabolic metabolic outcomes. So it's kind of the opposite of what we've been questioning so far, the controversial Blastocystis parasite concept. We see that actually having Blastocystis might be more beneficial than it was thought before. So taking that into account, we wanted to investigate what's going on in Blastocystis in Kent, in Southeast England. And so what we decided to look is IPS patients and see how this correlated Blastocystis presence. And I will demonstrate a few case studies that we've done. So if we got the ethics, we tried to recruit participants. This was a bit damaging because actually we got the ethics approved during COVID pandemic. So it was a little bit more difficult to recruit, but we managed to recruit a number of participants to look at this. And when we start investigating Blastocystis present, we identified that we have around 40% individuals had Blastocystis, which was something that is correlated with what we've seen across other urban areas. And when we've done the microbiome of this, we actually haven't seen much difference between the microbiome of healthy individuals and individuals that have Blastocystis as well. So kind of be stable. And there was no significant difference even in depending on the subtypes identified as well. When we've tried to group them by positive and negative or by subtypes, there was no anything statistically significant on the diversity impact either. This is to actually show you the compositional plots depending on the different subtypes. And as you can see, there is no strict pattern other than this subtype here. The only issue with that, because we only had one individual, so we cannot make major conclusions on that. What we've seen is that Blastocystis positive, which is in blue, has some connection with Procaecola and for example, which is a bacterium that is actually identifying more healthy individuals. So we can replicate what Nicolas Secada's group have seen on association Blastocystis present with healthy microbiome. What we also done, we investigate the metabolites, so certain metabolites from the stool samples to see if there are any metabolites that have specific signatures that could be correlated with the Blastocystis presence, so we can have them as markers. And we've seen some of them being recognized, but there was no significant difference between those in Blastocystis presence and absence either. And we are still exploring this even further. Among those individuals, we had a case study which we had a patient that had severe IBS symptoms, and he was treated with antibiotics. So we decided to actually collect samples from this individual more frequently. And we wanted to look at the study of antibiotics and the presence of Blastocystis as well. So When we screened for blastocystis on day 0, blastocystis was not identifiable. We saw him after the second day of antibiotics, third day, then blastocystis disappeared. And then after day 8 to up to 3 months later, blastocystis remained. When I said blastocystis absence, it means there was not be able to identify it with qPCR. Maybe the levels of blastocystis were much lower. And this was the same subtype consistently present across the 3 months after the cross. When we explored the presence of blastocystis during, when we look at the microbiome of this individual case, we did not see any clear pattern with blastocystis present, but we can see that this is a treatment of antibiotics, and you can actually see that the diversity of microbes is kind of reduced, which is something that it was expected. When we look at the metabolites of this, we can see some clear patterns on correlated with blastocystis presence or absence, and we're exploring this further. And we actually found some of these metabolites, for example, butyrate or valerate, to be correlated with blastocystis presence as well when the microbiome was coming to recovery. And what we are doing now is that we're exploring what's going on between blastocystis presence, the microbiome, and the metabolome. And what you see here is that you see, for example, this is the blastocystis presence correlation with microbes, and the darker the color is shown is whether they have positive or negative correlation with the metabolites present. And we start seeing some patterns correlated with blastocystis. As you can see here, this is red where this was blue, this is blue where this is red, which shows that there are some signatures there of the metabolic profile that could be correlated with blastocystis presence, and we want to explore this even further. Another case study we are doing is we are looking at the role in blastocystis in people going through cholecystectomy at Kent as well, and we collected samples on before the surgery, a couple of days after the surgery, and 6 months post-surgery to see if there is any connection or correlation with blastocystis. And we also do some metabolomics on the two samples. And what we've seen so far, this preliminary data, is that from the 6 patients, we have 5 patients were positive at least 1 point on blastocystis, and 3 patients gained blastocystis after surgery. And just to give you a pattern of what's going on, you can see that this is where surgery happened. So post-surgery, we start getting more frequently present of blastocystis. And I have a hypothesis on that as well. This is a metabolomics plot depending on whether it's pre-surgery, post-surgery, and after surgery, and you can see a different metabolic profile of the patients. And with the presence and absence of blastocystis, we can see a clear pattern. This is blastocystis negative and blastocystis positive. They are distinguishable. So we can actually get some metabolites to be correlated with the blastocystis present. And we are doing microbiome studies on that, and we want to explore this even further. So what I want to highlight from that is, first of all, on the antibiotics, is the treatment led to a significant decline in gut microbial diversity scores, followed by a recovery post-treatment. Despite antibiotic-reduced perturbation, blastocystis showed resilience, disappeared mid-course, and emerged shortly after the course ended. Antibiotic treatment caused shifts in the gut microbiome, including fluctuations in specific bacteria genera, such as Bacteroides and E. coli, which coincided with the presence of an absence of blastocystis. And we have alterations in metabolites that's showing that some specific metabolites can be correlated with antibiotic use, and this can be generalized more on the presence of blastocystis as well. And my overall point that I want to make is that from all these studies, what we've seen is that whether there's a patient or not, blastocystis is reemerging. So my hypothesis is that probably blastocystis is coming there to help somehow or correlated with the development of the microbiome to recover from a disease, from a surgery, or so on, something that actually affected the whole microbiome. This was obviously work done not by me. A lot of people work on this project. This is a few people that I want to acknowledge. Vasana, Abhi from Thailand, and Eleni, which was their supervisor, Satya, Rupen, and Jamie from my lab. And this is all my funding as well. And I want to go back to cost, because this is one of the reasons I'm here as well. I want to advertise what we are trying to do because of cost and because of all this I've been showing you today. As Marco mentioned, I'm part of the cost action Blastocystis Center of Health, and what we are aiming to tackle is a couple of important facts about blastocystis, how to detect and differentiate blastocystis, what is the role of blastocystis in the gut, can blastocystis cause disease or does it protect against disease, and how can we prove the knowledge of blastocystis. And there are a lot of misinformation about blastocystis as well that we want to tackle. One of the major aspects we want to do with the cost action is to assemble experts in clinical and public health sectors working on blastocystis or other eukaryotic residents of the gut in general. And this is where we want you guys to be involved as well. We want to harmonize wet and dry laboratory methods and consistent research methodologies on blastocystis. We want to develop a biobank and a data bank on blastocystis so people can actually expand their work even further. But also we want to develop models to examine blastocystis and see its role in the gut. So this is the whole concept of our action. If you want to be involved or if you want to get more information, take a picture of this. I will leave it for a few seconds. You can scan this barcode there, and basically you can be, if there's any interest of any of this aspect, from cell biology to epidemiology to any aspects of microbiology, microbiome, diagnostics, anything, please get involved with the action. Email me as well. And one of the things we are doing, and I want to emphasize a lot, we are trying to build, establish a baseline of blastocystis diagnostics in human, veterinary, environmental levels. We have a questionnaire circulating. I think Marco might have emailed a few people as well. He's involved with that. We want to get what is the knowledge about blastocystis in the various sectors, so we can find ways to actually improve and circulate some of the knowledge that currently exists and try to do a proper epidemiology study in Europe. So please complete the questionnaire, take a picture of this, forward to anybody that you think might be involved in all of this sector or be able to help. If you have any questions on blastocystis, I have the website here as well. This is our action website. We have a LinkedIn page, and we have a lot. We post almost every day on blastocystis. And this is my email as well. So if you have any questions about blastocystis, just email me directly. And with that, I will stop sharing, hopefully. The other thing I wanted to mention is that we also have an Instagram website. It's called Blastocystis. We have a little puppet. This is Blastocystis. And if you basically, you can follow us and you will get some tips and useful information about the Blastocystis and its role in health and disease. And thank you very much. Apologies for the troubles with the internet. I hope you were able to hear me.
Thank you very much. It was very, very interesting, very detailed. And maybe Marco, you can handle the discussion.
Okay.
If there are
Thank you.
any,
Thank you,
yes.
Tassos. And I don't know if there are some questions for Tassos. It was really a very interesting presentation full of information, especially for the majority of parasitologists that really do not deal with Blastocystis. And I think there is very, very few one. We have people from Tor Vergata University that know very well this subject. And I don't know, I can invite them to make a question to Tassos. Isabel, David.
If we have questions.
If you have questions.
Tassos. I talk with Tassos quite every day. I don't have any more questions for him. No, it was really interesting to see all the microbiome part. That is something that we are not doing here. For example, we just work with subtypes and molecular characterization. So it's a debate. It's an actual debate, really interesting to see if it's a pathogen or not, if it's helping the microbiome or not.
La sua opinione sul lavoro di...
No, già l'ho detto. Sì.
Just to mention, Isabel is one of our early careers or young investigators, and she's been very proactive with our actions. So we are very happy to have her along other early careers been promoting the Blastocystis cause action.
And this cost action, when is going to end, to finish?
We are currently on our third year, so we have 2 more years. So we have until November 2026.
Okay. And are you thinking about the submission of any Horizon project or thematic network? I think this should be a very important opportunity after building this network together.
So the first plan is to put a Marie Curie, hopefully in November this year. One of the reasons I'm actually coming to University of Trento in April to discuss this with Nicola Segata. Nicola is, obviously he's not a parasitologist, but he's brilliant. He's had so many ERCs and he's very well known in European Union. So having him as a partner would be amazing. And the other thing we are planning is we're trying to see if there is any specific call on microbiome or for Horizon Europe for an synergy grant or something like that. This is what we want to do. I built another thing I've done in December. Actually, I built a consortium of the big shots of Blastocystis within Europe to see if we can put guidelines of sampling, proper sampling collection for longitudinal studies. So if we can actually try to sample from across Europe and get a proper microbiome metabolome and Blastocystis study, that would be brilliant and answer a lot of questions, because everything that has been done so far, and I know everybody's doing their best with the finances that they have, including me, everything is sporadic. And we, one of the things I wanted to mention is that I contacted companies to talk about Blastocystis and see how if we can involve blastocystis in fecal microbiome transplantation and so on. And the answer I'm getting all the time, they're absolutely right, we don't know anything about blastocystis. So how you can put this in someone if you don't know what it's doing? And they're absolutely right. So we are 20 years behind than any other research. So we need to convince everyone that blastocystis is interesting, and people will need to explore blastocystis. That's why the purpose of the co-section as well.
And Tassos, I think that invite young scientists to join also because this year and the next there will be some very interesting training course.
Yeah.
And so it would be nice if we have more and more young scientists.
Yeah, and they
joining.
they can talk to Isabel if they have any question, because Isabel has been very, very proactive with that, and she knows what is going on. And the early careers are actually having their own group within the action, and they have frequent meetings, and they come to me with questions or events that they would like to organize, and I try to do my best to support them. So there is going to be a writing workshop in Portugal in September, which we will fund 16 early careers to go to that. And in 2026, we'll organize another writing workshop and also a hands-on workshop on wet lab and dry techniques on blastocystis. So people that they think they will be interested on blastocystis, if you haven't worked with blastocystis before, it doesn't matter. We will train you.
Bene, ci sono altri interventi, altre domande? Penso di no. So thank you again, Marco and Tassos. I hope you can also come to any SoIPa Congress or meeting we will have in the future. For sure, Marco has informed you that Roma will host the EMOP,
Yep.
the next EMOP, so for sure we will meet again. E grazie a tutti, grazie. E vi invitiamo al prossimo webinar del 24 di gennaio alle ore 17 e il nostro guest speaker sarà il nostro past president Fabrizio Bruschi, quindi vi aspettiamo numerosi. Grazie ancora Marco e grazie.